Okay, so big picture stuff. I'm just going to use this as a general orientation to where we're headed. Okay, it's a very busy slide. Okay, I know that because this process is busy. Again, because it's a process, it's much better to handle this with a YouTube or video stream. However, to talk about the details, we got to freeze frame it in 2 dimensions. So keep in mind that we'll be jumping back and forth between the 2 modes so that you can hopefully gain some understanding. And again, my suggestion is in all cases, you go back, you look at the YouTubes and things tonight so that you have a good understanding of what's going on. All right, so here we go. Take a look at this. Protein synthesis is basically going from DNA, right, which we know, what does DNA hold again? What's its main function? The genetic code. So you're taking that genetic code and you're making something useful, something visual, okay? A phenotype, if you remember that from genetics. That is what you see. Okay, so that's going from the code, the A to T to G to C, remember those 4 letters that everything on this planet shares, and making a protein out of it, basically, okay? And that's called protein synthesis, making proteins. All right, so we are going to go talk about the complex procedure whereby that occurs. And it's kind of neat, actually. I like this because it's kind of like a detective story. What happens is you take the code of life and then you transcribe it into a hidden message. The hidden message tends to be a strip of basically a thing called mRNA, messenger RNA. So think of that. You have a coded messenger RNA. Think of back like in the days of wartime, and they'd send a messenger with a code, a secret message. You'd try to make it back to the general or whatever to have a secret message. And so if it's intercepted on the way, the enemy couldn't get it and wouldn't be able to figure out or decipher what it is or translate it. And that's kind of what happens in this first step of transcription. There's a coded message going from DNA basically to mRNA. Okay, and they actually have words called codons. This word. So it's kind of a code, again, it's that secret part, okay? Then, once it gets to the general, which I consider the ribosome here, they translate that message into something usable. A readable message, in this case, happens to be a protein. Now, if you look up here, they don't have the word protein. This is kind of going back to the beginning of the year when we talked about different words for amino acid chain, and that word is polypeptide. That basically refers to the bonds between amino acids, peptide bonds between amino acids. So polypeptide means many amino acids, essentially, okay, which begets a protein, because a protein, remember, is made of amino acids, okay? So the translation part takes that coded message, those codons, and using anticodons, match the 2 together, and put the amino acid in the correct order. So what you're going to see is basically protein synthesis is taking the coded message here in the form of A to T's to G's to C's, right? Because that's all you see in DNA, and transcribes them into a coded message. And we're going to see there's a little change there, and we're going to talk about that in a second. A new rule, because there's no T's in RNA. Remember that way back a couple days ago? So that transcribed message has a U in there to kind of throw the enemy off. And it comes down here into the ribosome as translated into an amino acid change. So the sequence here in the DNA is preserved by the sequence here. It's a unique recognition that this code really is the same as this code. So the sequence of amino acids is coded for back here in DNA, and that's why the protein comes out perfectly, assuming no errors occur in the process. Okay? So that's kind of the big picture. Hand up. Yes, sir.
So is it like all the what's it called? Like the amino acids and everything are like all by themselves, and then they get like changed into DNA things?
Yeah, very similar to the process of DNA synthesis, where you have those DNA nucleotides floating around in the cytoplasm, and the DNA polymerase, remember, grabs them and puts them in the correct order. Okay? We're going to see a very similar process here. You have another enzyme, and thanks for the segue, called RNA polymerase, which does essentially the same thing. It takes free-floating, basically RNA nucleotides, and if they're binding to the tRNA, transfer RNA, we're going to talk about that tomorrow more, and it puts them, grabs them out of the cytoplasm, puts them in the correct order based on the sequence that was provided by the mRNA. But yeah, pretty much. Okay? Yes, ma'am.
Do the 3 to 5 ends make any difference?
No, not really here, because what we're talking about, and thank you for the segue there, we're talking about a gene. And a gene is a piece of DNA that is usually embedded within the DNA. And let me just pop this up and use our strand up here. Okay, so this DNA here, let's just say that this is remember, DNA can be hundreds of thousands of bases long. So let's say this is one piece, let's say it depicts one gene. Remember, we haven't really defined gene yet because it's more of a genetics, but we will now, okay? A gene is just a physical part of the DNA that codes for one specific polypeptide. It used to be one gene equals one protein, but what we found now is sometimes, in some cases, there are several polypeptides that get put together in the Golgi body, actually, to synthesize into one protein. So, in the last 20 years anyway, I mean, in your lifetime, it's always been this way. So, basically, one gene begets one polypeptide, and in many cases, probably 90% of them, one gene equals one protein. So the gene is a stretch of A's to T's to G's to C's, and you're going to see there's going to be a start and a stop that says, okay, start here, stop here. It turns on the process that we're going to talk about now. Now, let's give you big picture stuff here. Remember, in your body, every cell in your body has the same DNA message. Okay, so that means every cell in your body has the ability to become every other cell. So the question, I guess, is, why aren't they? Why is a liver cell just a liver cell and not an eye cell? Why can't certain cells become other cells? Well, that's the real, $10,000 question that's out there. We kind of understand the process, but it has to do with the turning on and off of genes. And what we find is as an organism becomes more advanced, like our cells, that on switch, that regulation that turns a gene on, can't really be turned off. At least we don't really know how yet. But in simpler organisms, like did somebody here do a current event on the worm? We study simpler organisms because, for example, planaria, you can cut it in half and it can regenerate its entire body, brain, everything, and survive. In fact, you can cut it. There's all kinds of studies that show that happening. So lower organisms have the ability to totally turn on genes kind of at will, whereas in our case, it doesn't quite work that way. So you cut off your arm, you don't regrow an arm. That isn't an ability to regenerate. But we can constantly regenerate skin cells, hair cells, nail cells. So certain cells are able to do that, whereas others aren't. Okay? So keep that in mind. So this whole idea of protein synthesis, the regulation of that's a whole other field we'll talk about at the end. But the process is pretty much the same. And that is turning on the transcription and translation of a section of DNA to create a polypeptide/protein. Okay? That's the picture. Okay. Yes, ma'am.
If we can recreate skin cells, then why do people get fake skin put on them to cover up something? Because I know that's
Yeah, that's a huge. Yeah, that's a.
my grandpa has it doing it right now, so there's a machine on him.
Is resynthesizing cells.
Yeah.
That's tremendously new. Yeah, they have a printer essentially that prints skin out now, and we can regrow skin in tissue, because the way it used to happen is you'd have to have skin transplants. And so you have to cut a big swath of skin off and then take from here like you put up here. And the problem was you'd have obviously scarring and stuff, and it was a terrible thing, and there's all kinds of chances. So now we're able to, starting with the whole cloning thing, and I think somebody, I can't remember, all these current events are running together, but some of you did presentations on taking cells, and before they differentiate and the genes are turned on, you can really let them become any cell. And that's cloning. That is where we're at now. And that's fascinating because you can do hearts and you can do organs and tremendous things without the rejection issue. So anyway, last question, got to move on. Go ahead.
Since on the skin replacement thing, my friend fell in fire once and had to get skin on her leg, and they took it from her butt. Is there a reason why they use that skin from that part of the body as opposed to others?
Because you can't see it. The scar can be covered up. Yeah, I had a friend that burned, I think I told you the safety thing, a friend that burned himself tremendously, 80% of his body, usually he would have died, but anyway, same thing, he had graft, but he only had so much grafting that he could do in his legs, because you can, put pants on and not see it, but the face and stuff, so that, it basically has to do with cosmetics more than anything else. Okay. From what I understand, I'm not a doctor, okay? All right, here we go. If you remember back in the day, 2 days ago, way back in the day, we gave you some rules, and the rules were the binding rules of DNA, and that's Chargaff's rules. Remember A to T, G, C, that complementary rule. So in other words, when DNA is synthesized, if you have a template strand, A, the corresponding anti-parallel strand is going to have T on it, etc., right? We went over that. Now, we also at that time I just said, okay, I'm going to throw this out, but we're not going to deal with it until today. So today we're going to go back and revisit that, and I'll show you the practicality of the new rule, and that is in DNA there is no T, all right? There's only U in DNA. And so what happens is when DNA is processed transcribed and translated, the RNA switches out the T's for U's. And the thought process is that the enzyme involved, RNA polymerase, the enzyme that makes RNA, recognizes the U's, not the T's, so that it doesn't jump over to a DNA and start transcribing it. Okay, so that's the thought. Okay, and if you remember back in the primer sequence in the RNA, they changed the RNA to DNA. This is how they did it. They just took the U's out and substituted T's. So that was kind of working backwards. So the rule is, and I would write this down somewhere. You might want to write on a piece of paper on your notes, is instead of when the enzyme RNA polymerase transcribes the message from DNA, it does it by inserting a U in place of T's. That's basically the only change. Everything else is the same. If there's a G, it's going to make a U. Or C, I'm sorry. If there's a C, it's going to make a G. If there's a A is going to put a Q in. If there's a T, it's going to put an A in. Okay? So the only change in RNA, other than the changes we talked about, the base and all that stuff, the change in the actual nucleotides is adding the U for uracil instead of the T for thymine. Yes, ma'am?
Can you repeat what you said we should break down again about the enzyme DNA?
Hold off. We'll get to that in a second. That'll be part of the PowerPoint. That's coming. I just threw that in there as a bonus. Yes?
What happens to the time when it switches out time for the uracil?
Well, in this case, it doesn't really switch out. I was talking about back in DNA synthesis. You remember they showed the primase coming in, and then it changed it over to DNA. Don't worry about that. I was just kind of giving an FYI thing. Okay? All right. So again, this rule is the new base pairing rule. It's not really new, but it's a different one for RNA. If you look up here, there are 3 types of RNA. We're going to learn about those as we go through this process. So as we go through this process, I'll tell you what the M, the T, and the R stand for, and we'll take notes as according. Okay? So let us begin. All right. So transcription. Transcription is the first part of a 2-step process called protein synthesis. So if you want to title your notes, protein synthesis, step A, transcription, go for it. Okay? Again, protein synthesis is making a protein from DNA. Part A is going from DNA to RNA. And I would like you to put mRNA there. My colleague made this up, and I would have put an M there, but so it's more specific, okay? So transcription is basically going from DNA again to mRNA. Okay? So how does that happen? Well, just FYI, just to go back and refresh, basically your nitrogen base in the DNA strand is what really determines this whole thing, right? Again, the sequence of the A's, the T's, and G's, and C's, that's the starting point, and that is the part that's important, okay? Just to let you know, there are 20 types of amino acids, and because that's going to be the final product, amino acids, okay? And if you look on your handout that I gave you, and this is a chart, and your book has one of these too. This is the universal code And what that means is every organism on the planet, as far as we know at this point, with a couple exceptions, again, organisms, viruses really aren't considered a living organism, but they kind of a little wacky rule. There's a couple other new